we will discuss about the liver function tests. They are together called as hepatocyte function panel, where 7 tests are done. The liver is situated at the right side of our abdominal cavity. It shows various functions, including the synthesis of protein like albumin and blood clotting factors. It helps in heme catabolism. It helps in storage of fats and biles. It has several other functions related to metabolism. To produce energy from food is another major function for liver. This is the picture of normal liver and this is the picture for cirrhotic abnormal liver. By liver function tests, it is easier to detect any sort of disease presence within our system, as well as different types of disease can be treated, detected, and the extent of the liver dysfunction that is related to the disease can also be detected. So, different types of disease, degree of disease, as well as the presence of disease can be detected. Not only that, the follow of a particular treatment can also be done with liver function tests. The prognosis, outcome of any disease or treatment in the future can also be detected by liver function tests. It may be synthetic function, hepatocyte injury related, or cholestasis related. So, synthetic function means the functionality, the activity of the liver is related. So, it will be correlated with albumin production. It will be correlated with the clotting factors production. When we are talking about hepatocyte injury, it will give rise to the maintenance of hepatocyte cellular integrity. Aspartate transaminase, alanine transaminases are actually related to these tests. Cholestasis, the obstruction related to biliary tract. We know that biliary tract is correlated with the liver. So, if any obstruction occurs over there, that can be detected by bilirubin, alkaline phosphatase, as well as gamma glutamyl transpeptidase activity detection. Liver function tests are non-invasive, specific, as well as sensitive. So, the presence of any disease, the extent of disease can be followed. But there are some of the disadvantages of the liver function tests. Always they are not specific as well as sensitive. So, we have to do all the tests together. Suppose in case of hepatitis A, that can be detected by the elevated level of alanine as well as aspartate transaminases. But in case of hepatitis C, it cannot be detected. Though liver is dysfunctioning, that cannot be manifested at those serum alanine transaminase as well as aspartate transaminase levels. A slight elevation of the liver function tests can be observed in case of fatty liver. Fatty liver is actually observed by obesity as well as the presence of diabetes mellitus. Apart from these, there are other diseases also which give rise to the abnormal liver function tests. Viral hepatitis A, B, C and other viral diseases, autoimmune disease, hepatoma, Wilson disease where extra amount of copper is getting accumulated, hemochromatosis where excess iron is getting absorbed, celiac disease, Crohn's disease. So, there are so many diseases where the abnormal liver function tests can be observed. Not only diseases, but also some of the medications can also give rise to abnormal liver function tests. What are they? Antibiotic like tetracycline, cholesterol lowering drugs, painkiller drugs, diclofenac, they can give rise to abnormal liver function tests showing the damages of liver. And students, chronic alcohol intake can give rise to the elevated level for liver function tests. Now for the liver function test, the blood will be collected from the vein. The patient must be 10 to 12 hour fasting in condition, and from the whole blood by centrifugation, we can collect the supernatant as serum for the following tests. Synthetic liver function tests. It includes albumin production as well as production for blood clotting factors. Together they are called as coagulation panel, including prothrombin test as well as international normalized ratio.
Ma'am, but why is it called synthetic liver function test?
By these tests, we can assess the activity of the liver, the synthetic power of the liver. As we know that the liver synthesizes all of the protein related to bone, blood, etc. So, it is called the synthetic function assessment of the liver. Serum albumin is the major part of blood protein apart from globulin that can be assayed very easily. By kit, bromocresol green dye is dissolved within the buffer, which along with the albumin gives wonderful color, can be measured at 620 nanometer by spectrophotometer. As standard, bovine serum albumin is given in the kit. So with different concentration of bovine serum albumin according to their absorbance or OD at 620 nanometer, this particular standard curve can be made. So by putting our optical density for the serum albumin, we can easily detect the serum albumin that is present within the serum. Normal reference level for albumin is 3.5 to 5.3 gram per deciliter. It includes prothrombin time as well as international normalized ratio. For blood clotting, some of the factors are needed, the protein factors which are synthesized by the liver. Factor 7 is very important factor, but factor 8 is not synthesized by the liver. So it follows the vitamin K dependent extrinsic or common pathway for coagulation cascade. Now you can follow the blood clotting time by taking the serum in the citrate anticoagulant, and we can mix the tissue factor containing thromboplastin. We will record the time for the blood coagulation in seconds by photo-optical or any sort of electromechanical device. So that will give you the time for clotting. The normal ranges lies in between 8.8 to 11.6 seconds. If any deficiencies occurs by the liver damage or lack of synthesis by the liver, especially the factor 7, that can be manifested in the clotting time. It will delay. International normalized ratio, it is actually proposed by the World Health Organization, WHO. International normalized ratio is the ratio in between the prothrombin time test divided by prothrombin time normal, that is related to the international sensitivity index. So this particular international sensitivity index is correlated with international reference preparation of thromboplastin, which is actually corrected by correction factor, and here the international reference preparation is 1. So ISI is 1 for the normal. So by comparing the INR as well as PT, you can correlate the functioning of liver, on what extent the clotting factors are produced by the liver. It actually gives you the idea about the cellular integrity of the hepatocyte cell within the liver. So two enzymes are there, aspartate transaminase as well as alanine transaminase. Why are they called transaminase? Because they add amino group from one amino acid to another alpha keto acid to produce another amino acid. Sometimes this aspartate transaminase is called as serum glutamic aspartate transaminase, and these ALT or alanine transaminase can be called as serum glutamate pyruvate transaminase. So, what is the full form of SGPT?
Ma'am, it's serum glutamate pyruvate transaminase.
Very nice. So, let's talk about the principle for assay. It is according to the IFCC method proposed in 1991. From alanine and oxoglutarate with the help of the enzyme pyruvate and glutamate is getting produced. This particular pyruvate can be converted to lactate by the help of lactate dehydrogenase enzyme, where NADH is participated with the pyruvate, which is getting converted to NAD+. This NADH is absorption at 340 nanometer. So, by reduction of 340 nanometer OD, we can have the idea about the enzymatic activity, especially the enzymatic activity for SGPT. The interesting thing is that, you know, the SGOT, SGPT, they have several isozymes present in cardiac muscle and in other muscle. So, the elevation of these two enzymes are not always related to the liver function, though they are taken as the traditional tests for the liver functioning, but they are not actual yet, because in case of myocardial infarction, in case of other muscular problem, their activity may be increased. But it is also true that by liver injury, they may be spilled from the liver to the serum. So, elevated level can be obtained. Now, the working table, the enzyme reagent as well as standard is taken. As standard, you can take any standard enzyme that may not be provided by the kit. From these standard enzyme, you will have the idea about the extent of reduction of the NADH, which will thus help you to construct such calibration curve. And by following the standard curve, you will have optical density of your serum preparation. So, by putting your OD value, that is especially the decrease in OD value, you can have the assessment of serum alanine transaminase. Because you know SGPT is also called as alanine transaminase. The normal value for alanine transaminase or SGPT is around 5 to 40 unit per liter. Actually, these value varies according to the different laboratory where which type of quality control is taken. So, what do you mean by SGOT?
Ma'am, SGOT is serum glutamic oxaloacetic transaminase.
Okay. So, SGOT stands for serum glutamate oxaloacetate transaminase. It may also be called as aspartate transaminase. From aspartate and alpha ketoglutarate, oxaloacetate and glutamate is getting produced. Can you see the reversible reaction is going on? Now, the oxaloacetate is a kind of alpha keto acid. According to Rittman and Frankel, they were actually proposed this method at around 1957. These particular alpha keto acid is reacted with 2,4-DNPH. What is that? Dinitrophenyl hydrazine to get dinitrophenyl hydrazone derivative, which has red color, can be measured at 510 nanometer. So, this working table shows you that how much substrate will be added, how much DNPH and NaOH, because the presence of NaOH will thus help to produce the red coloration. And you know that always the blank is made by the water. So, from the standard curve, we have taken pyruvate as standard. Can you tell me why? Any idea? Okay, I am giving you the answer. Though pyruvate is not produced by the enzymatic reaction, it is oxaloacetate, but it readily transforms to the pyruvate. So, 2 millimole per liter pyruvate is taken as standard, from where the series of standards are produced, which gives the different OD values by reacting with 2,4-DNPH. Now, what you are getting by enzymatic activity? Again, pyruvate. Now, it is the standard method, that is 1 millimole pyruvate produced per minute corresponds to 1 unit per liter enzyme activity. So, aspartate transaminase activity in unit per liter versus absorbance will provide you a standard curve. Now, the product produced pyruvate by your serum enzyme will be plotted here to get the enzymatic activity. The reference level, the normal range of this AST lies in between 8 to 20 unit per liter. So, we must think about the linearity, we must think about the preservation of the kit reagent that must be kept at 2 to 8 degrees centigrade, and always the reaction will be compared with the quality control. Biliary tract is attached to the liver. So, cholestasis is related to biliary tract obstruction, any inhibition in the bile passage. So, these are correlated with the bilirubin alkaline phosphatase as well as gamma glutamyl transpeptidase activity detection.
Ma'am, what is cholestasis?
Okay, exactly mean liver actually transform indirect bilirubin to the conjugated direct bilirubin. This particular bilirubin is actually passing through the biliary tract towards the duodenum. If any obstruction occurs over there, it actually designates the biliary tract dysfunction as well as liver dysfunction. So, that is actually called cholestasis. You know that the higher content of bilirubin in the serum is manifested as jaundice, the yellowish pigmentation in the skin, in the retina, etc. And in case of neonates, you can observe the neonatal jaundice. So, how can you monitor them? You know what is indirect bilirubin? So, from heme catabolism, first the bilirubin that is produced, they are not solubilized within the serum. They are actually making adapt with the serum albumin. They are referred as indirect bilirubin. This particular bilirubin within the liver is getting conjugated by the liver enzyme with the glucouronic acid, and this particular conjugated bilirubin is passed through the biliary tract towards the small intestine. Some portion of the bile that can be absorbed by the liver as well. So, if we get the increase in the conjugated bilirubin, that means the dysfunction related to the hepatic as well as the biliary tract. They may be termed as the intrahepatic as well as post-hepatic dysfunction. But if indirect bilirubin is increased within the serum, that is correlated with the rapid hemolysis. The direct bilirubin can be assayed with the help of diazotized sulfanilic acid, which gives rise to azobilirubin. At alkaline pH, it has blue color, which can be measured spectrophotometrically at 607 nanometer. Again, total bilirubin can also be counted, where by putting accelerating agent, that is caffeine benzoate acetate mixture, which will thus help to solubilize indirect bilirubin, which ultimately give you the azobilirubin. Again, blue in color at alkaline pH, which can be measured by spectrophotometer at 607 nanometer. Now, by eliminating the value of direct bilirubin from the total bilirubin, you will have the idea about the indirect bilirubin content, and from there you can assess whether this is post-hepatic, hepatic or pre-hepatic type of jaundice. Now, working table, by following this particular working table, actually the bilirubin tests are done. You know that cysteine reagent is actually help to stop the reaction. Now, we can make this particular calibration curve with respect to different concentration of the bilirubin and their corresponding absorbance or optical density at 607 nanometer. Now, putting your value for the direct or total bilirubin OD, you can have the idea about their content, their concentration present in the serum. Let's talk about their range. In case of adult, the total bilirubin is ranged from 0.3 to 1.3 milligram per deciliter. The direct bilirubin content is 0.1 to 0.4 milligram per deciliter and indirect is 0.2 to 0.9 milligram per deciliter. In case of infants, it is quite higher because rapid hemolysis occurs over there. You know, their globin chain is very rapidly changed to adjust the environmental oxygen pressure. So actually their bilirubin content varies from 1 to 12 milligram per deciliter. So actually phototherapy done to remove their indirect bilirubin content. Alkaline phosphatase are found at the inner wall for biliary tract. If it is getting disturbed, they are spilled outside and they can be found within the serum. Now these alkaline phosphatase may be related to the bone and placenta. So in case of teens and kids, you can find the elevated level for alkaline phosphatase because bone formation occurs, though that is related to the functioning of the liver or the adjoining biliary tract to the liver. The artificial substrate paranitrophenyl phosphate is taken, which is dissolved in the buffer at pH 10. Now, upon the action of alkaline phosphatase, it gives rise to paranitrophenol and inorganic phosphate. This particular organic compound paranitrophenol has absorption at 405 nanometer. It is yellow in color. So, if you prepare the standard curve for different concentration of paranitrophenol that is also provided by the kit, and if you have the corresponding absorption or OD, you can prepare a standard curve. Am I right? Yes. Now, putting your value for the paranitrophenol that you are getting from your serum preparation, you can have the idea about the concentration because you are getting their OD. Yes. You are just getting the product formation from the serum and you are plotting the value here, so you will get their concentration. From this particular concentration, you can calculate how the milligram per ml paranitrophenol produced within the incubation time is actually the alkaline phosphatase activity, which can be presented in unit per ml or unit per liter of the serum. Now, what is the standard normal range? That is 20 to 70 unit per liter. So students, I have told you different types of liver function tests. If you are getting any abnormal value, say for alkaline phosphatase, you have to detect other tests. But what I have told you, in some cases like aspartate transaminase, alanine transaminase, they may not always correlated with the liver functions or rather liver dysfunctions. So the true liver function tests are albumin as well as coagulation panel, including prothrombin time and international normalized ratio. They will actually tell you the prognosis factor. That means what sort of treatment is done, how far the liver function can persist within the serum, that can be manifested as the liver dysfunction or not, can be assessed by albumin, because the clotting factors as well as albumin, they are solely synthesized by the liver. But traditionally the AST, ALT or SGOT, SGPT are done by the laboratory as LFT or liver function tests. So you must go for the cross checking with respect to other enzymes.